Hi, Michelle Glass here. Welcome back to another episode in our Chapter 8, Lecture 8 series. And our topic here is finally mitosis. Now, mitosis is, of course, asexual cellular division for eukaryotic cells. Remember, asexual means without sex, and in this case, that means no genetic diversity. So we're making clones, and the purpose of mitosis then, this type of cell division, is for growth and repair. Now we know that there are 4 key stages, and we're going to go through those in a lot of detail. So we have our memory device, PMAT, to help us remember prophase, metaphase, anaphase, telophase as our 4 phases. And this is the process of dividing the nucleus. So the key thing that we're looking at is the dance of those DNA chromosomes as we go from 1 cell into 2 cells. So let's get started. Let's start with an overview. So here we're looking at the cell that's in interphase. So this is before DNA replication. This is before mitosis has occurred. And just to simplify my drawing, we're looking at just 1 homologous pair. So this cell would have a diploid number of 1. That's not right. 1 pair, 2 total chromosomes. All right. Our homologous pairs or homologous chromosomes match up with each other based on their length and also based on carrying the same type of genes, but they're not identical to each other because the cell is inheriting 1 of each copy from each parent. So the egg is contributing 1 of our number 1 chromosomes, and the sperm is contributing 1 of our number 1 chromosomes. So this means that they're not identical to each other. I give the example here of them carrying the gene for hairline. So I have a straight hairline. The other type of hairline that you can have is the widow's peak. So it comes down a little bit at the V kind of shape. And this is a human characteristic that is controlled by a single gene, and there's different forms. And so this is how we're talking about they're carrying the gene for hairline, but there's different forms. So maybe the mother is contributing the straight hairline and the father is contributing the widow's peak. Okay, still in interphase, we need to replicate our DNA because ultimately we're going to be making 2 cells and we need enough DNA to go into each one of those cells. So DNA replication, remember, is happening in S phase of interphase. So again, before mitosis. Once you have DNA replication, that's when those sister chromatids appear. Remember, sister chromatids are identical to each other and they are attached at that location called the centromere. And then we're going to see that this cell goes through the stages of prophase, metaphase, anaphase, telophase. We'll go into all of those processes here in just a few minutes, and we end up with 2 daughter cells. These daughter cells are identical to each other, and they're identical to our starting cell. Again, you're making clones. The purpose of mitosis is for growth and repair. So you want to make sure that you're repairing with the same type of cells, so genetically identical. This is the signature of being asexual reproduction without sex. Okay. So I'm going to do a series of drawings. You can also draw these in your notes. If you don't want to do that, maybe you get your textbook and take a look at the artwork that's provided there, or just write out in words. So I'm going to have my parent cell, and it's going to have a diploid number of 4. So this time I'm going to be drawing 2 pair of homologous chromosomes. Now this is our cell before any DNA replication has happened. This is our cell before mitosis, so we're in interphase. During interphase, you have your S phase. That's where you get DNA replication, so that's when you're going to start to see your sister chromatids forming. They're going to be attached at the centromere. Now here's a key thing to notice. During interphase, the DNA is in its relaxed state called chromatin. So I've drawn it here as condensed chromosomes just so we can see it visually and keep track of it, but it would really be all relaxed in that nucleus. Okay, now we're actually ready to start mitosis. So we're going to go through PMAT, prophase, metaphase, anaphase, telophase. So first stage is prophase. Now notice that three major things have happened. Number one, and this is not in a particular order, we have our spindle apparatus forming. So in animal cells, we have centrioles where those spindle fibers are coming off of those centrioles. We see that the nuclear envelope will disappear and the DNA is going to condense to make those chromosomes. So now we actually do see those chromosomes when you're looking at the cell under the microscope. Now we're ready for metaphase. In metaphase, those spindle fibers have attached to those centromeres and pulled those chromosomes so that they line up in the middle of the cell. Now they can line up in any order. So I'm showing you here the two long and then the two short, and I have my greens together and my oranges opposite. You can line up with both of the oranges together, long and short, and both of the greens together, long and short. So that's just kind of random how they line up there. My next phase is anaphase. In anaphase, those spindle fibers are going to contract, separating the sister chromatids. So now they're going to opposite ends of the cells. And then we get telophase. Telophase is basically a reversal of prophase. So we see the spindle apparatus disappearing. We see that the nuclear envelope reforms, and we will see that DNA relaxing back into chromatin, although I didn't show that in my drawing. I wanted you to see that these two cells are going to look the same way as that original cell. You can see too that the process of cytokinesis is starting. Cytokinesis is the division of the cytoplasm in animal cells that involves the formation of the cleavage furrow. So we'll talk more about that in just a minute. Okay, so I learned this kind of hand dance on YouTube, so I thought I would share it with you all. So you can bring your hands together like this to represent prophase. So your fingers are representing those condensed chromosomes. If you have those condensed chromosomes, then we can also remind ourselves that our nuclear envelope is going to disappear and that spindle apparatus forms. Then bring your hands together to represent the chromosomes lining up in the middle for metaphase. Pull them apart to remind yourself of the separation of sister chromatids that happens in anaphase, and then form two fists to represent the two new nuclei that are formed during telophase. So you have prophase, metaphase, anaphase, telophase. So that can be just a real easy type of thing to do to help you remember those stages. Okay, so let's look at that process of cytokinesis. Mitosis, remember, was the division of the nucleus, the DNA material. Cytokinesis is the division of the cytoplasm. So that means all of the other stuff inside of that cell has to split. Now this occurs differently in our animal cells from our plant cells, and the reason is because plant cells have a cell wall and animal cells do not. In animal cells, you get that cleavage furrow forming, bringing those membranes together until they connect and then pinch apart and separate. So they basically just pinch in two. In the case of your plant cells, they have to actually construct a new cell wall to separate. So that kind of primitive cell wall that's being formed is called a cell plate. And then once that cell wall is finished, then you have two identical separate cells. Okay, that's it for now.